So we decided, welcome back to the activity metabolomics and mass spectrometry short course. I decided to get going right away today because it's we have quite a bit we want to cover. And actually, I even wanted to briefly cover a question that came up while we were sitting waiting to get started about actually having access to some of the instrumentation that we have. So Bill Webb, who's going to be talking in a little bit, he basically does all the training in our open access lab and our advanced open access lab. And that allows individuals to have access to relatively straightforward instruments like the single quad instruments, but also triple quad instruments and more sophisticated systems. So, and Bill and Elizabeth and Corey are always willing to train people on whatever technology that they need to get trained on to perform the experiments. So that being said, today's presentations are going to be a little bit more intensive in terms of the theme of last week. So last week's theme was that we covered some of the basics, and then we jumped into more advanced aspects of the technology. And since last week it was actually so successful, and there was a lot of positive feedback on how we did that, we decided to bring in an additional cameo speaker who I'll mention in a few seconds. So as you all know, we're hosting this at Scripps Research. This is where my lab is, and where most of the speakers that you're going to hear from today are here at Scripps Research. This is one of the images, and this is the room that we're located in right now. So the on-site group is relatively small, as you can see from the size of the room. This is not a live image. The live image is we have roughly about 12 people here now or so. But we did have a substantial number of registrants. I think it's now hovering somewhere above 3,200 or so. And it was nice to see such a great showing last week. And apparently, I just heard that the showing is actually quite high today as well. So thank you all for attending. Today, these are the primary speakers that we're going to have talking over the course of these 4 weeks, or the 4 Tuesdays. Today, it's going to be myself, Elizabeth Billings, who I introduced last week, Myrna Tabia, and Bill Webb, and they'll be speaking about the proteomic side of the technologies that we have, and also the targeted metabolomics. Next week, we're going to go more into the untargeted metabolomics, designing those experiments, and also some of the software aspects of those. We're using XCMS and Melker. And as I mentioned last week, one of the cameos, and Juliana Ivanisevic she's one of the panelists that's watching me right now. I can see her on there, and welcome, Juliana. We appreciate your visiting us from the University of Lausanne in Switzerland. I know it's going to be about 8 o'clock when you give your presentation, so we really appreciate it. She's going to be talking about population scale metabolomics, which I understand there's a lot of interest on this particular topic, because we're going to be speaking initially on a very fundamental level of how we do these targeted sort of analyses. And Juliana is going to take that into hyperspace, with not only looking at many more molecules than we're going to mention, but simultaneously looking at huge numbers of samples as well. So Juliana is definitely considered one of the leaders in this field, and I'm looking forward to her presentation at the end. Since we are doing some proteomics today too, a short basics on proteomics, John Yates has graciously offered to talk as well. And I'd be surprised if any of you in the audience don't know John. John is a professor here at Scripps Research. He is considered one of the originators of modern proteomics technologies. He came up with the original paper back in 1994, introducing basically LC-MS technology to identify proteins. And ultimately, he designed a sequest program, which has become industry standard. and is available and widely used throughout the world still, even 30 years later. So I was really happy that John agreed to give this presentation, and he's another one of those hyperspeed presenters today. He's going to, we're going to start off with very basics of proteomics, and he's going to jump into how he is using proteomics to study disease mechanisms. Okay, so last week we went through a wide variety of things, including insource fragmentation. This week, we're going to start off with a proteomics segue with Myrna and Elizabeth. And then we're going to further focus on the targeted metabolomics, again, from a fairly basic standpoint. This will be after John gives his presentation. And then we're going to end with Juliana's presentation on population scale metabolomics. Just briefly, proteomics, a definition that I think is fairly useful is proteomics is a discipline in which a complete set of proteins within cells or an organism is analyzed. I like to think of metabolomics as simply the same thing, except replacing metabolomics for proteomics and metabolites for proteins. And essentially, I like to think of metabolomics as being functional proteomics. It really is giving a sense of what's happening within the cells and what the proteins are doing. So they're very much intertwined with each other, and the technologies are also very intertwined with each other. As you'll see today, much of the mass spectrometry technologies that we're using are very similar, if not identical, except for how we ultimately analyze the data that's being generated. And so it's worthwhile, and there's actually a lot of Nobel Prizes that have been given with respect to protein technology and protein understanding proteins. Beadle and Tatum is one of the earlier ones, one gene, one enzyme, sequencing and synthesis of a protein by Wegener in 1955. Sanger, of course, which in terms of Sanger sequencing, many of us are familiar with, and in 1980. And the one I always like to refer to, because basically Fenn and Tanaka both won Nobel Prizes for mass spectrometry-based protein analysis. And again, they demonstrated that you could use electrospray and soft laser desorption ionization very effectively on proteins. And it became more apparent over time that not only could you use these technologies for proteins, but also very easily for small molecules as well. And that's why we primarily use these technologies for metabolomics, at least in our lab. Klaus Biemann is probably one of the really early innovators of using mass spectrometry for sequencing, and he did that as early as 1958. William Hensel came up with had the first paper, him and a number of other investigators, including John Stoltz at Genentech, and describing protein identification with enzymatic digestion to identify proteins with mass spectrometry. And as I mentioned, John was the real innovator behind what is now what is basically one of the cornerstones of proteomics. LC-MS based automated identification of proteins. And as I mentioned, John is also here at Scripps. And I'm also very proud to say that one of the other really major innovators in proteomics, especially activity-based protein profiling, is Ben Cravath. And he actually developed this technology back in 1999. And for some of the results that have come out of this, he recently won the Wolf Prize. So it's really an honor to have colleagues like John and Ben here. John has also won many awards I don't know, John, you're on there right now. So would you say, if you shake your head yes or no, it's about 20 awards or so? Is that about accurate? Yeah, he's saying yes. Okay, with that, I'm going to have Elizabeth get started on our very rudimentary aspects of proteomics, and then Myrna's going to follow up.
Thanks, Gary. So for proteomics, we're going to talk about two main approaches we take for these analyses. The first of which is intact protein analyses, where we're trying to determine the intact molecular weight of the protein and also looking for potential modifications on the protein. So to do this, we use two techniques, which Gary alluded to in his timeline. The first of which is electrospray ionization. So when you do the AMMS of intact proteins, we'll take the protein in a mass spec compatible buffer, usually avoiding any sort of interfering ions from detergents or other incompatible components. And we're going to charge that by electrospray ionization. In this image here, I have an ESI TOF pictured. We can either have an electrospray source paired to a TOF or quad detector. And in both cases, we have great sensitivity. So when we're analyzing these intact proteins, we're typically getting analysis of proteins at the micromolar concentration, but you could detect even lower depending on your experimental setup. When we're using a TOF detector, we also have great resolution and accuracy. So in the context of proteins, this is especially important when trying to determine small mass modifications. So on a protein like BSA that's about 66 kilodaltons, we can observe modifications at the tens of daltons level using an ESI TOF instrument. The other important aspect of ESI ionization is that it's easily paired to an LC system. For intact proteins, this can be used for on-column desalting. So if you have your protein in something like PBS, you can wash the salts off during the analysis. You don't need to have it outside your buffer, and you can remove those salts from interfering with ionization. You can also separate proteins from other proteins with this solution or other small molecules that may be present. The other important aspect of ESI ionization is that we're going to get a series of multiply charged peaks for each protein. So in this example, I have the charge envelope for egg white lysosome. So you can see a series of multiply charged peaks here. And from these multiply charged peaks, we can determine the intact mass of the protein through the process of deconvolution. In this case, about 14 kilodaltons. This charge envelope is relatively simple because it's a relatively small protein. The other important thing to note here is we're only seeing transmission of multiply charged ions for the intact protein. We're not typically seeing any in-source fragmentation like we observed last week with the small molecules. This is due to the large degrees of internal freedom that we see in peptides and proteins, and they can take on those charges without having issues of in-source fragmentation. If we take a larger protein, the charge envelope becomes much more complicated, and it'd be quite difficult to process this data manually. However, we can still deconvolute to determine the intact mass of 66 kilodaltons. And this is enabled by the fact that deconvolution is completely automated in the software. So you can go from collecting your raw data to determining the intact mass of your protein in a matter of seconds. The last thing I'll note here is if you look at the x-axis of these, you'll see that the M/Z range is about 2,000 to 3,000 daltons max. If we did not see multiple charging of the protein, we would not be able to observe these in this small scan range. The other approach that we take for ionization of proteins is MALDI. So through MALDI MS of intact proteins, in this picture, I have a MALDI TOF instrument. MALDI is not compatible with LC because instead of injecting the samples through a liquid chromatography system, we're spotting them on a stainless steel MALDI target. And we're spotting these with a specific matrix. And because we're spotting them, we're typically using less than a microliter of samples. So you can use a very small amount of protein, about at the same concentration of a micromolar range. And we select a matrix depending on the compound of interest. So for an intact protein, we're typically using synapenic acid matrix, but if you're analyzing something else like a smaller peptide or polymer, you would need to select a different matrix for optimal ionization. The way that MALDI works is you're ionizing that co-crystallized matrix and protein together and looking for that charge. The other thing important to note, since there's no LC here, you need to be very mindful of salts when you're spotting the samples, as they may hinder ionization. The other aspect of MALDI that we take advantage of when analyzing intact proteins is its high scan range. So on this picture here, you'll see that we're seeing the singly and doubly charged peak of BSA. Unlike before, we were seeing a series of multiply charged peaks. So this is what is typical of MALDI analysis of intact proteins. We'll see a singly and doubly charged peaks. And we can take advantage of the fact that the scan range for the M/Z goes up to 200,000 to 300,000 rather than that 2,000 to 3,000 M/Z we were seeing before. The other thing of note here is that this technique is a bit lower in resolution. So on this same BSA where we were seeing modifications of the tens of daltons, we can see modifications of hundreds of daltons here. And the peaks may be quite broad. But MALDI is still great to utilize if you have a small amount of sample and you want the results relatively quickly. But I would say if you're looking for modifications, ESI would be more recommended. So that encapsulates a very basic overview of how we do intact protein analysis. I'm now going to pass it over to our next speaker, Myrna, who's going to discuss enzyme-based protein characterization and how we use that for protein identification and post-translational modification analysis. Thank you, Elizabeth. So just like Elizabeth mentioned, our second approach for proteomics is enzyme-based protein characterization, which includes the digestion of proteins into small peptides using a specific enzyme. And since we're on the topic of protein identification, I'd like to take this opportunity to mention that we're very happy to have the proteomics pioneer, John Yates, here with us today to discuss the topic of studying the mechanism of disease shortly after me. So for protein identification with proteomics, we mainly accept samples that come in two forms. The first one being in gel, and we prefer in gel since the protein in the gel band gets purified. And we prefer SDS page gel bands that can be visualized well by either Kumasi or silver stain, which indicates that there's a sufficient amount of protein in the gel band for the protein identification analysis. We also accept samples that are protein samples that are in solution, and we typically ask for 100 femtomoles to 1 picomole of proteins, or 5 micrograms of proteins. And we strongly suggest that these samples are free of any detergents or unnecessary buffers that are incompatible with mass spectrometry in order to avoid any interference with the data results. So now that we have the protein sample, we can proceed with the protein identification analysis, which includes reduction, alkylation, and trypsin digestion, which digests the protein by breaking down the amino acid protein sequence into small peptides. And then these digested peptides are analyzed by the nano-LC that is coupled to the Q-exactive, which also has an ESI, resulting in MSMS data. And that MSMS data is searched against the predicted fragment ions from theoretical digestive proteins contained in a protein database, such as NCBI or SwissBrot, and using the proteomics search engine software. And there are many tools that can be used to utilize this type of data. One of them being the original protein search engine software, which is Sequest, that was actually developed by our next speaker, John Yates, about 30 years ago, and is still in use today. So after you run the search, you will the software will generate a list of identified proteins, with the top protein result being the most abundant protein. And I want to pass it to Gary now, which will introduce our next speaker.
Great, perfect timing. So actually, given the number of speakers today, that you really handled that very well. And one of the reasons for us doing this was just to give some individuals in the lab a chance to speak in front of an audience. We didn't quite anticipate that the audience would be as large as it is, but it's just has heightened everybody's awareness as a result of doing these presentations. So it's nice, actually, we're doing these presentations in front of a relatively small audience, so that helps quite a bit. So I think we now are going to switch over to John. Actually, I think thanks again, John. I think roughly this is a 20-minute introduction to John Yates.
So John, you can start sharing your screen now whenever you're ready.
Can you hear me?
Yes.
Okay, good.
Let me turn off my screen.
How's that?
Perfect.
Great.
And I've got 20 minutes, is that correct?
That's correct.
Okay, good. All right, so what I'd like to talk about today is just some work that we've been doing for quite a while to try to better understand mechanisms behind disease. In particular, we've been trying to study cystic fibrosis and try to understand why the genetic modification to the CFTR gene causes the protein to not mature properly and fail. And I like to show this picture from, I think it's David Goodsell, who's also at Scripps, which kind of illustrates that cells are packed full of proteins, lipid bilayers, and all sorts of things. And they're not just bags of water with proteins floating around in them. It's actually a quite congested environment. All right, so cystic fibrosis the gene was discovered about 1989. It's one of the most common lethal genetic childhood diseases, affects about 1 to 4,000 newborns in the US. It's the main clinical phenotype, so you start to accumulate mucus, which obstructs airways and digestive tubes. You get recurrent infections, inflammation, respiratory failure, and life expectancy prior to the drugs being developed, it was about 15 to 40 years, depending on what kind of mutations that you had. And so the mutation in turned out that CFTR is a anion channel that regulates chloride homeostasis, and when that's no longer functions, you start to accumulate. You don't get the water on the surface of the cells, and you start to accumulate mucus, which gets rather thick and becomes a great environment to cultivate bacteria. It causes these infections, recurrent infections, which eventually, over time, the inflammation associated with that starts to destroy the tissues. So about 70 to 90% of CF cases are caused by deletion of delta F508, and that causes the CFTR protein to misfold. So as I mentioned, the CFTR gene was discovered through genetics. It was discovered in 1989 by Lap Chi Choi, Francis Collins, and Jack Reardon, and postdocs and grad students that were involved in that project. It made rock stars out of Lap Chi and Francis Collins. Francis Collins went on to become director of the Genome Project and then director of the NIH. In 1992, it was discovered that it was a temperature-sensitive mutation, which was a clue that that protein folding issue might be a problem. It took many years to get a structure for the protein. Crystallization just totally failed, primarily because the protein has some disordered regions in it that makes it difficult to get a well-ordered structure that's required to get a crystallization. So for the wild-type protein, cryo-EM was used in 2017 to get a structure, and then it took 5 more years to get a structure for the delta F508 version of the protein. Drugs were eventually discovered by phenotypic screening about 26 years after identifying the gene. And this helped with the most common mutation. And the reason why phenotypic screening was used to try to discover drugs is that people just didn't understand the mechanism behind the failure of the protein to mature, why it was misfolding, and so on and so forth. In the intervening years, discovered with genome sequencing becoming relevant and becoming more common, about 1,400 different mutations have been discovered. And it's not uncommon for someone to have, for example, delta F508 allele, and then have some other mutation in the other allele that causes the disease to occur. If you're homozygous for delta F508, you're pretty sick. So as I mentioned, drugs were discovered. This was since this is considered to be an orphan disease, not enough patients, that the major pharmaceutical companies really didn't have programs to deal with trying to identify drugs. And so the Cystic Fibrosis Foundation funded Vertex to come up with drugs to treat the disease. And then one of the first drugs they came up with was this VX-770 drug in 2012. And that didn't work very well for the delta F508. It's a potentiator, so it causes the channel to open and stay open. And so it treats a certain not so common mutation that some patients have. And then in 2015, Orkambi was developed. This resulted in a potentiator as well as a corrector. And so the VX-809 drug was intended to be a corrector, so it helped correct the fold. At least it was thought that it was helping correct the fold of the protein. And in 2018, there were a couple more drugs, but it wasn't until 2019, 2020, when Trikafta came out, that there was really this really had an impact on patients. And in fact, it's really changed a lot of lives in a number of ways. And the story in the Atlantic describes one woman's absolute change in life by the drugs that were developed for this. So certain drugs can really have a huge impact on people's lives. And so we started by about 20 odd years ago trying to understand what it was about why this protein was not maturing, was not getting made, and so forth. And it becomes a sort of a model system in a way for membrane proteins which don't mature and get out to the cell surface to do things. And so we thought by looking at protein-protein interactions, we could try to understand aspects of that. And then that led us into looking at post-translational modifications. And this is we started doing this. I started asking questions about structure prior to the crystal structures coming out. And so our hypothesis was that if we looked at the protein-protein interactions in the wild type protein versus the delta F508 protein, we would be able to learn something about why the protein wasn't folding properly and what proteins were involved in that process and so forth. And so we hypothesized that there was a disease-specific interactome that we would be able to discover. And so we had developed this workflow all centered around mass spectrometry, all centered around the things that were just discussed, isolating proteins, then mass spectrometry, and then database searching, as well as all this other informatics that was performed in order to figure out what were specific interactors versus non-specific interactors, and then trying to figure out the networks of proteins that were involved. This was done with shotgun proteomics, which was just described, where you just digest the protein mixture and run it through the mass spectrometer, and then identify all the proteins and quantitate the proteins that are present. And so one can easily describe what we found based on this graph. The proteins that were associated with the wild type version of the protein only are up here in the top, and those that are associated with the delta F508 are down here in the bottom, and the differential interactions are kind of in the middle. And so you can look at the gene ontology categories of proteins that were discovered, and they make a lot of sense: ER quality control, degradation, trafficking, folding, and then plasma membrane interactions. And so you can see that there's clearly a disease specific interactome that is present in the protein. And so we tried to understand which of those interactors were critical for rescuing the protein. And so we looked at 52 novel interactors. We did knockdown experiments, and then we just used Western blots as a way to try to figure out whether the protein was now maturing to get out of the cell surface. And we found that 48 of the proteins that we had identified influenced CFTR maturation, and they all belong to about 7 different subnetworks of proteins. So we then tested which of these will actually rescue the function of the proteins. And so we did these crude electrophysiology experiments where we could measure whether the channel was functioning properly. And we tested about eight different proteins, and we found that all the ones that we tested, with the exception of one, actually rescued the protein using this Usine chamber experiment. And if you look at the sort of gold standard for rescuing the protein, the 28-degree temperature shift, so you lower the temperature, the protein folds enough to get out to the cell surface, and you treat with forskolin, which is a stimulator, and then you treat with genistein, which is a potentiator. And then to make sure that it's the CFTR ion channel that is open, you treat it with a CF inhibitor. And that should shut down the current, which is exactly what you see when you do this experiment. And so when you knock down certain proteins, you can see that you indeed do get recovery of the ion channel function. This one particular protein down here, you see when we knock that one down, we don't get any restoration of channel function. And so what we discovered doing the interactome is that there were a lot of kinases and phosphatases and so forth that were involved in the interactome. So that was telling us that PTMs might in fact be important. And we went in and we did the PTM analysis using the sort of traditional mass spectrometry experiments. And we found that indeed there are a number of modifications to the protein. So we found 40 different PTM sites, about 70% of them were new. And we found 21 fossil sites, 6 methylation sites. And as you might imagine, the Delta 508, there were a lot of ubiquitination sites since the protein is being targeted for degradation. And we found that some of these modifications clustered in what we were calling hotspots. And we found that phosphorylation was very important. We did several different kinds of experiments, TMT experiments, Silac experiments, targeted experiments to try to figure out the nature of the phosphorylation and whether there was we found that there were 3 different sites very closely together, but we wanted to know whether there was single phosphorylation that was required or whether it was double or triple phosphorylation. Turns out it's just single phosphorylation at one of these 3 sites that was important. And it turns out it was a casein kinase 2 alpha, 1 consensus phosphorylation motif, and that allowed us to do a whole bunch of other different experiments. But we also looked at whether phosphorylation was required for traffic, was an indicator of CFTR trafficking success. So we looked at different mutations, some which were trafficking competent. In other words, the proteins were getting out to the cell surface and they were functioning, and versus those without trafficking impaired, which is what the Delta F508 version of the protein is. And we found that phosphorylation was a signal that the protein was mature and that it was going to exit and get out to the cell surface. We found that if we take the wild type protein and we use an inhibitor of that particular kinase that we thought was doing the phosphorylation, we find that we gain back 59 of 71 interactions which are normally specific for Delta F508. So by inhibiting the phosphorylation, we find that the wild type protein starts to look more like the Delta F508 protein, and it doesn't get out to the cell surface. And so we think there's a PTM code for CFTR maturation. So it requires that we have phosphorylation on one of those 3 sites, plus a nearby lysine site is methylated. And if that happens, then the protein is determined to be correctly folded and it gets out to the cell. It starts to exit the ER and gets out to the cell surface. If the phosphorylation if it's not properly folded, you don't get phosphorylation. What you do is you get ubiquitination, blanketing, bracketing that phosphorylation site, and then the protein gets targeted for degradation. And so we curious, why would that be? And so it turns out we had done an experiment many years before where we looked at the wild type NBD1 domain. It was produced in a cell line or insect cell line where the site wasn't going to be phosphorylated. And we did cross-linked it with HSP90. And turns out that the site where the phosphorylation is, it was an interactin site with HSP90. And we also found that in our experiments that the amount of interaction between HSP90 and the NBD1 domain goes up by a factor of 4. And so what we believe was happening, which is what we described in this 2012 paper, is that the HSP90 binds to the NBD1 domain and it forms a chaperone trap, which then gets targeted for degradation. And we believe that a phosphorylation, we haven't done the experiment, but we believe if phosphorylation is present, we don't get that HSP90 phosphorylation trap. So we started asking questions about structure and how to look at the structure inside the cell rather than outside of the cell. And so what we wanted to know was what does the protein look like when it's in the cell and when it's interacting with all these different proteins and so forth? Is there a difference between what we might see on the outside? And it turns out that there are some mass spectrometry-based methods that have been developed over the years, probably over the decades, where people have used hydroxyl radicals as a way to label the surface accessible regions of proteins. And of course, the traditional method that's been around for a long time is HDX, hydrogen deuterium exchange. There are some fluorophore methods, there's chemical labeling methods that Richard Vacher has been developing. And then of course, is the protease sensitivity assay, which the Picardi lab in Switzerland has developed. And I first became aware of these methods when Mike Fitzgerald came into the sabbatical in my lab. What Mike wanted to do was take his Sprox method and take it proteome wide, which he did while he was on sabbatical in my lab. And he published this really nice paper, which I think really was probably one of the first attempts to look at the 3D proteome, in vivo 3D proteome. So we developed an alternative approach which incorporates quantitative analysis into this. And so we use formaldehyde together with cyanoborohydride to label the intact cell or tissues. We have a paper that is just accepted in Nature Communications where we're labeling an entire mouse using this strategy. And so we label first with one weight of formaldehyde, and then we digest up, and then we label another with a different weight formaldehyde. And so we get a differential quantitative measure of what's surface accessible and what's not. This provides us with a way to look at the conformations, in particular, of proteins that are inside the cell by doing this method. And when we look at the CFTR protein and we compare it to the cryo-EM structure, we get really good agreement between the cryo-EM structure and the proteins. But one of the tricks they had to play in order to get the cryo-EM structures is that they removed the regulatory interface element that's deleted, and that's the place where the modification, the PTM code goes on the protein. And then they have also removed that to get the delta F508 structure, they also added a stabilizing mutation into it. But we found that with the structure that was available versus what we were finding in the CPP experiments, we found that there was good agreement between what we saw and what they saw. So one of the first experiments we did is that we did 4 different labelings, 4 different biological replicates. We did light and heavy, light, heavy, and then we did a label swap experiment. So if you're familiar with SILAC experiments at all, you do a label swap experiment to make sure that the light versus heavy labels are not affecting the reactions at all, the observations that you're seeing. And the wild type protein are the dots in blue, and the dots in red, the squares in red are the delta F508 proteins. So you can see that there are some significant differences between the surface accessibility of these lysine residues within the protein. And one in particular at 273 is very pronounced, where in the wild type protein, you see it's not very accessible, but in the delta F508, it's very accessible. And so that caught our attention, of course. And so we followed up on that, and it turns out that K273 is in this ICL2 domain of the protein, and that domain interacts with the NBD2 domain. So this is nucleotide binding domain 2. This is intracellular loop 2. And so that's an interaction that we think we suspected from our data was very critical. And so if we take VX770, which is a drug that causes the protein to be in an activated state, constantly in an activated state. And so the channel is open. And when we treat with that and we look at this position of K273, K273 is not accessible at all. So it's deeply buried into that pocket within the NBD2 domain. And normally the wild type protein is mostly buried in that domain, and the delta F508 is not buried in that domain at all. So there's no interaction between the ICL2 loop and the NBD2 domain. And so if we now look at some other conditions, so we take this is the wild type protein over here. And so if we now take compare the wild type protein versus the delta F508. So this is the fully activated domain, fully activated protein here. No treatment, DMSO treatment, then treatment with VX809. If you remember from my earlier comments, VX809 is this chemical chaperone that's supposed to help the protein fold. And what you see when you look at this data for K273 is that it basically does nothing to K273. The gold standard for rescue of the delta F508 is this temperature shift experiment. And you can see that the temperature shift causes that K273 to be a little more buried in the NBD2 domain. It gets buried enough that it can recover and mature and get out to the cell surface. And so basically this VX809 drug doesn't seem to be doing much of anything. We do have a theory for why it seems to work. And so this allows us to come up with a model for the conformational changes. So when the protein is activated, the K273 is buried in the NBD2 domain. And when you get partial 273 exposure, this might be when it's at the plasma membrane, perhaps during activation. And when you have full K273 exposure, it's inactive. And maybe during transport. So if you look at Trikafta, we find that Trikafta actually does pretty well in terms of moving the K273 domain in terms of where it should be. It's quite just like the delta the temperature shift experiment, and so that suggesting that it's actually changing the conformation of the protein enough that it's becoming activated. All right, so good timing. Just to summarize, so CPP, this chemical protein painting reveals vastly larger conformal space for CFTR than molecules and was captured by the cryo-EM. This dynamic movement of CFTR domains is critically important for trafficking and activation. Movement of this ICL2 loop is impaired in misfolded CFTR mutants, and knowledge of this ICL2 interface status could help develop better CF drugs and drugs for CFTR mutants not currently treatable. And the people that have done all the work, in particular, Sandy Pankow has been doing this work for 10, 15 years. Casimir helped with a lot of the PTM experiments, and they both worked on developing the CPP method as a way to look at protein conformations. And that's the rest of the lab. And thanks.
Yeah, thank you, John. As we transfer over to Bill Webb and Elizabeth, I just had a quick question for you about what your presentation. The one thing I was surprised that you didn't cover, and I'm be curious, and you can start transferring those. So one, of course, this fibrosis is very much characterized by lots of disulfide bridges, and you didn't really talk about that at all. And I was wondering what role the mass spectrometry technologies that you developed help decipher some of the complexity of those interactions.
So we didn't look at disulfide bridging primarily because it didn't really seem to come up in the studies that we had been doing, and we had to be very careful about what sorts of experiments that we were doing, because CFTR is not a highly abundant protein in the cell. So there's really only a few hundred copies in cells. And so, in fact, this is what stymied us early on in our studies 20 years ago, is that mass specs just weren't that sensitive. And so we were having to grow up many plates of cells in order to get enough material to do experiments. And it wasn't until, probably the Orbitraps came along that all of a sudden we had sufficient sensitivity that we were down to 1 or 2 plates of cells in order to do these experiments, and things really exploded from that point on. But we have not looked at disulfide bridging because it just hasn't seemed to come up in terms of the questions that we wanted to ask next. To answer your question?
Yeah, that makes sense, because I think the first part of the whole process is the ordering of the proteins, or the misordering of the proteins, and then that, my understanding was that stimulated ultimately the formation of the disulfide bridges, but the initial part, it sounds like, is what you were focusing on.
Yeah, we also, one of the other questions people always ask is, did you look at the carbohydrates, the glycans on the protein? And of course we have not looked at that because it hasn't really been come up as an important question so far. At some point, we'll probably want to look at that, because there's probably significant differences between the glycans on the wild type protein versus the delta F508.
Well, thanks a lot, John. And we're going to jump into the next section, which is
Sorry,
actually. So I guess yes, you're not shared any longer.
How do I do this?
I think you're all set.
Am I unshared?
You are unshared, and we're back to metabolism and metabolomics, and just going to briefly discuss a little bit of background of metabolism. There's lots of metabolism-related Nobel Prizes. I'm just going to touch on a few of them, so we can get right into the next section of talks. So the Warburg effect was one of the original ones. Discovery of vitamin C and the catalysis of fumarate in and the Krebs cycle, of course, and then mechanism of regulation of fatty acid metabolism by clock and line. Again, from a metabolism perspective, in metabolomics, I would also put Fen and also perhaps to a lesser extent Koichi Tanaka, but certainly Fen in terms of the development of electrospray for for biological type molecules, especially metabolites. And even though it was given largely for mass spectrometry, it's had such a broad impact in a wide variety of areas. The paper I always like to refer to, in addition to the book that I mentioned last week, which was the Woolley book on anti-metabolites, I always like this particular paper because it's probably one of the first papers that we found, and I say we, it was actually Bill Wyckoff in my lab, he's a former postdoc, who found this paper, and I think it best describes what we're doing today, but it was done back in 1966. And it was basically describing, as the title says, gas liquid chromatographic procedures for separating out wide range of metabolites in urine and tissue extracts. And this, I think, is perhaps what would be considered the origins of developing the technologies for the types of things that we're trying to do today. And ultimately, one of the bottlenecks in what we're trying to do today is identify what these molecules are and statistically assess the differences between them accurately. One thing that I touched on last week is that we still, surprisingly, and this is over almost 60 years later, we're still challenged with the very reliable identification of these molecules. So it's not been a trivial process. And again, this sentence from the abstract of this paper largely describes what we're trying to do today. And again, this was roughly 60 years ago. I typically break metabolomics down into two separate categories. One is targeted, and that is what we're going to focus on today. Next week, we're going to focus more on untargeted based approaches and how we design the experiments and also how we do the comparative analysis and ultimately the identification of the molecules that we're observing. So as I mentioned earlier, we're going to start off with a very basic aspect of targeted analyses that Bill and Elizabeth are going to describe, and then we're going to jump into Juliana Ivanisevic, who's going to describe a really very broad scale in terms of the number of molecules that she's looking at, but also the number of patients or individuals that she looked at for these analyses. So with that, I'll give it to Bill.
Thanks, Gary. As Darren went to next week, when you're looking at untargeted metabolomics, you need a high resolution instrument to give you the high accuracy mass measurements, because finding out exactly what these compounds are can be quite time consuming and difficult. On the other hand, you know exactly what you're looking for. Say you want to look at amino acids, for example, or compounds in the TCA cycle, or just a novel compound that you want to do a time course. The type of instrument you use for that is a triple quadrupole mass spec. Triple quadrupole mass spec consists of an ion source, initial quadrupole, Q1, a collision cell where fragmentation occurs, sometimes also referred to as the second quadrupole, then the third quadrupole, and then the D detector. There are lots of different experiments you can do on a triple quad, but if you want to measure how much you have of your compound in a sample, the experiment we would do is a multiple reaction monitoring experiment. In this case, you would set the initial quadrupole to isolate on a particular precursor M over Z value. In this example, either a molecule M over Z 210 or 350. You would then send those into the collision cell where they would then fragment, and then you set the third quadrupole to isolate on a particular fragment ion. So in these examples, only compounds that have a precursor M over Z of 210 or 350 and a corresponding fragment ion of 191 or 226 make it all the way from the source to the detector. How is this helpful? Well, we're going to look at some data collected with a single quadrupole of a serum extract. And this was collected in single and selected ion monitoring. We're only scanning for a single M over Z value. In this case, we're scanning for an M over Z value of 426. Researcher wants to look at this compound in his serum. Issue is, in a serum sample, it's a pretty complex mixture. So there could be more than one compound there that has M over Z of 426. And if you want to try and quantitate and get accurate quantitative information off this, it can be difficult. It could be a lot of error can be introduced due to the presence of isobaric species. So we can take the same sample, same serum extract, analyze this on a triple quad using MRM method, where we set the first quadrupole to isolate on the parent M over, or the precursor M over Z of 426. We'll fragment this in the collision cell, and then only let a particular fragment ion, in this case M over Z 196, pass through, and the upper trace on the single quad neatens up quite nicely on the triple quad. So you can get a lot better quantitative information on the triple quad using the MRM method. And this also allows you to weed out all the things that you don't care about, only focus on the things that you do care about. So because we set the triple quad to only focus on specific M over Z values and not scan full ranges of M over Z values, our sensitivity is boosted by about a factor of 100. And because we're employing this MRM method, where we're looking at a particular precursor and a particular product, this really enhances our selectivity as well. So this also results in nice linear calibration curves, which further improves your quantitative data. So since we're talking about biological samples here, we have to do some type of extraction first before we can introduce them to the instruments. And no extraction procedure is going to be 100% efficient. So we take this into account by the addition of internal standards. So an internal standard, take into account losses during prep, okay? We'll want to make sure that this compound is not going to be present endogenously in the samples that we're analyzing, because that can introduce error as well. We want these compounds to be chemically similar, but differ in mass. And if you look at the two compounds at the bottom here, on the left, you've got tryptophan, and on the right, we've got a deuterated version of tryptophan. Isotopically labeled compounds are the best internal standards because they're chemically identical and they just differ in mass. However, it can be difficult to come up with a deuterated sample sometimes. It can be cost prohibitive, or you're working with a novel compound and there is no deuterated version. In this case, you want to find something that's going to be chemically as similar as possible, so it's going to behave as closely as possible to the compound that you're measuring, but it differs in mass. This will take into account not only matrix effects during the extraction procedure, but also ion suppression when there's a lot of other compounds being sprayed in the spray chamber at the same time, which is common when analyzing complex mixtures. So that's a brief summary of triple quadrupole work. We're now going to pass on to Elizabeth, and she's going to discuss how this is applied to other compounds. Okay.
Thanks, Bill, for that brief overview of MRMs. So a few different applications of how we typically use MRM approaches for our lab. The first of which is pharmacokinetics. So if you're looking to do a PK study on a drug or prodrug, you can use MRMs to measure the levels of that compound over a time course and calculate your PK. We also can use this to develop metabolic panels or pathways. So in this image here, you could, for example, develop a panel of the TCA metabolites if you want to see how those are changing in your samples, or for example, if you want to look at a panel of amino acids. We can also take this to apply to any sort of targeted metabolite of interest. So as long as you have a standard for that compound, a method can be developed using LC-MS, and you can quantitate those compounds into your biological samples. So now I'm going to give two specific examples of some recent studies we've done in the lab applying MRM technology. The first of which is an amino acid metabolic panel. Someone came to us, they wanted to look at plasma from mice that were fed a serine glycine free diet versus a controlled diet. And they wanted to look at how those amino acid levels varied between these two groups, especially with that deprivation of serine glycine. So for this approach, we used helichromatography based on the chemistry of the compounds to look at the panel of amino acids of interest. So the next table I'm going to throw at you has a lot of numbers in here. This is the actual MRMs that we developed. So in the leftmost column, you'll see the list of the amino acid compounds. You'll see that each has a unique precursor ion. And then you'll see two columns here labeled quant ion and qual ion. So these are the two fragment ions that we're looking at in Q3 for that particular precursor. And the first one, the quant ion, is typically the most abundant ion we're seeing. And we're going to use that to actually quantitate the compounds. So when Bill was showing those curves of where we're measuring underneath that area to quantitate, that's what we're using. We also monitor for a second fragment called the qual ion to further specify to make sure we're looking at the compound of interest. So in this case, you'll see that those are all kind of varying depending on the different structures of amino acids. But in this way, we can create a full quantitative panel of amino acids, all done with one run, one injection on a helix column. Another example we looked at, which actually ties in quite well to our lectures, I didn't actually know the example that John Yates was going to discuss, was samples of bronchial alveolar lavage fluid from cystic fibrosis patients. So in this study, they wanted to look at various lipids in these samples. And because we were looking at lipids, we used a reverse phase chromatography technique. So in this case, we were looking at some specific LPCs, PCs, and DPPC. And this is a quite simple analysis where we're going to target specific lipids of interest. But in Juliana's approaches with population metabolomics, she would be looking more broadly at classes or larger groups of compounds. This table, similar to the first one, is the MRMs for these half a dozen LPCs and PCs. But what you may notice, while they each have a unique precursor ion, we're looking at the same quantifier ion for all the compounds and then two sets of qualifier ions. This is because of the way that lipids fragment. We're actually looking at a fragment of the head group of these lipids. So even though those quantifier and qualifier ions are the same masses, they're the same over M over Z. Because we're fragmenting from that particular precursor, we're still specific enough to quantitate these lipids of interest. So those are two projects that we did on this. I'm going to pass it back to Gary to introduce Juliana.
Great, thanks. And Juliana, actually, so I guess Bruno will just transfer it over, but Juliana Ivanovicevic, she is a former postdoc in my lab, and after she finished, she went and joined the University of Lausanne as a professor there. And she's been doing really a lot of different types of interesting work, and oftentimes with a very large focus on the technology, but also having one foot in the door of actually applying it to biological type questions. And kind of like last week when we had Erin give a presentation, and it was I think it was particularly enjoyable because of her dynamic nature. I don't think Juliana is going to be any different from that. One of her energy has been one of the things that she's always been known for. So we're looking forward to hear this. And again, thank you, Juliana, for being willing to present at 8 o'clock in the evening in Lausanne.
Thank you very much, Gary. I'm really happy to present and to share my work. So I will first try to share the screen. I hope this will be successful. Is it fine?
Yes.
Yes.
Okay, perfect. And you can all hear me well?
Yes.
Yes.
Okay. So hello, everyone. Many thanks, Gary, for this very nice introduction. And really thanks, to all of you, to the entire organizing team for your kind invitation. So as I already said, I'm really very much happy to actually have this opportunity to share the work of my team on unleashing the potential of lipidomics for the population health research. And so I really hope that the rest of my presentation will be as fancy as this title. So I will first start by briefly introducing you the problematics, and then I will really focus on the analytical platform we have actually built for the lipid measurement at a very large scale, and this from the perspective of the sample size, but also the lipid coverage. And I will then, at the end, finally really share with you some preliminary results we have obtained so far through the profiling of the Lausanne population, which nicely demonstrate the sex specificity and the high individuality of our circulatory lipid signatures. Sorry, I'm just, oops. Okay, it works. So why lipidomics, and why today actually the time is right to integrate lipidomics as a powerful phenotyping approach in the epidemiological studies? Well, ever since the Framingham Heart cohort started, so now more than 70 years ago in Massachusetts, and this very tight association between the circulatory cholesterol and the risk of heart disease has been revealed, the lipid measurement has actually become and remained the mainstay of cardiometabolic risk assessment. I'm sorry, but oops, my slide change is a bit slow. The keystone of clinical chemistry diagnostics are these very well-known bulk components of our plasma lipidome, including the triglycerides, free fatty acids, and cholesterol, whose total content is used actually as an indicator of dyslipidemia and has been integrated into the risk prediction models, and this usually on the top of the blood pressure. And please consider here that these all these different lipid classes and many others I will talk about today are actually incorporated within these lipoprotein particles that you can see the image of on my slide. Or they can also be conjugated to the albumin, which actually enables their transport in the blood as a really highly hydrophobic metabolites that are not soluble in the aqueous plasma. And beyond these very few actually lipid measures that are available in clinical routine, it's obvious that today, with the mass spectrometry-based lipidomics approaches, we can measure many more lipid classes, and that in many more details. And here you can actually see the results from one of the first interlaboratory effort, which was done in order to characterize the NIST plasma reference material. And you can see on this scheme that more than 20 different lipid classes with an impressive diversity of lipid species could be detected in human plasma. So here actually each line represents one lipid species, and if you go down, you can see actually its corresponding analytical consensus concentration, which was established during this interlaboratory effort. So overall, more than 1,500 unique lipid species were actually reported in human plasma at that time, so back in 2017, and this using multiple technological platforms, multiple methodologies, but also modes of analysis. And during this past 2 to 3 decades, these different mass spectrometry lipidomics approaches were actually applied to hundreds of case controls, so mainly cross-sectional studies, in order to understand how the lipid plasma composition changes and how it can be actually associated with different health and disease states. And indeed, many different lipid classes, and this far beyond only triglycerides and cholesterol, have been found to be significantly positively or negatively associated with our cardiometabolic health, which is here represented by the cardiorespiratory fitness as one of the most integrative measures of our organismal function, but also one of the very well-established predictors of all-cause mortality. And indeed, depending on their biological roles and also the involvement in different metabolic processes, such as the energy production and storage, insulin signaling, regulation of the immune response, different lipids can actually have indeed the beneficial or the deleterious effect on our cardiometabolic health. And importantly, this lipid effect is not only class dependent, is actually species dependent. And today it's very well known that the structurally closely related species, even different stereoisomers, can have different biological roles and therefore be associated with distinct metabolic consequences. And one of the most well-known examples of the lipids with a deleterious effect on our cardiometabolic health are specific species of ceramides, which have actually been identified to be diabetogenic and atherogenic, and this especially when they accumulate chronically, so over longer periods of time, with the excessive calorie supply and the inflammation. But however, and despite of the fact that the ceramide score has already been introduced in several private clinics worldwide, the ceramides, as this is nicely said as you can nicely read from the title of this famous review written by Scott Summers, they still remain the devils we don't know enough about, and this especially compared to cholesterol. So there is really an obvious need for further fundamental biochemical studies, but also the longitudinal population and the clinical intervention studies to gain further mechanistic insights about their mode of function. And this at least before the ceramides really become our new cholesterol. And this is also true for many other lipid species, which have also been significantly or found to be tightly associated with actually cardiometabolic risk. So we need the detailed actually lipid profiling across many different model systems is really needed to further understand the role that these individual lipid species play indeed in the disease onset, development, and the progression. And importantly, we also know that the models, predictive models with more species will also potentially have the more predictive power. And this is one of the main reasons why we recently got involved in several epidemiological, but also clinical intervention studies. So mainly the longitudinal study of the Lausanne population, so named COLAUS, which was established at the University Hospital of Lausanne, but also the complete health and heart cohorts, which were created actually already 10 years ago by the sports medicine doctors at the University of Basel. And here our main idea is actually to collect the critical amount of this lacking steroid and lipid data that can nicely complement the data that have been acquired through the clinical characterization and also the genotyping. So why is this? Well, mainly because these lipid profiles, these comprehensive lipid profiles, actually represent the endophenotypes or the phenotypes at the molecular level, which are genetically less complex compared to these endpoint disease outcomes, but equally heritable. And as such, these profiles can actually help us to explain the biology behind these revealed associations between the genetic variants and the endpoint clinical outcomes. And this is also nicely supported by the fact that the genetic loci, which are associated with the blood lipid levels, are also associated with the cardiometabolic disease traits. Sorry. One yet another very important point I really want to make here is the need for the perspective, for the studies actually with a prospective and the longitudinal character. And this mainly because the cross-sectional case control studies are not able to identify the prognostic markers, nor to infer the causality between the lipid levels and the clinical outcomes. And to be able to really acquire this valuable lipid data in as reliable manner as possible and with the highest specificity possible, we have first focused on the optimization of the lipid analysis pipeline for the large-scale population phenotyping. And here our main idea was really to minimize the technical, so analytical variance, to have really the maximum power, to allow for the maximal power in the statistical analysis later on. And this is also one of the reasons why we went ahead with the development of a targeted approach, or in this case, the high coverage targeted approach, so which we kept still at the omics scale, not to compromise the discovery. So why targeted approach? Well, simply because we wanted to measure, as also want many biologists and clinicians today, several hundreds and up to thousands of known and very well-characterized lipid species in a straightforward way with high robustness, high sensitivity, and a wide dynamic range. And we went with this decision also because of these still unmet, untargeted challenges, among which I will cite only two. So firstly, the limited sensitivity, and secondly, really the time-consuming annotation all over again of this very well-known and well-characterized lipids. So this goes also for the polar metabolites, which can be actually quantified in a straightforward way. And with the development of this high coverage targeted approach, we also in a way wanted to bridge the gap, as you can see from this scheme, between the two extremes. So on one side, this targeted approach with which we usually typically focus on one pathway or one class of metabolites of interest, and the untargeted approach on the other extreme, with which we simply tend to detect as many polar or lipid metabolites as possible. And to really warrant the application of our analysis pipeline in these large-scale studies, we have went ahead with the single-step extraction, in this case using isopropanol, mainly to really warrant or maximize the reproducibility and the ease of automation. So if you want to know more about it, don't hesitate to check in details. I mean, more details are available in the publications I have cited below in the slide. But briefly, please consider that before applying this single-step extraction using isopropanol, we have really extensively tested its performance against the biphasic extraction protocols, which are commonly used in the lipidomics community. And then for the lipid quantifications, we have decided to go ahead with a high-leak method, so coupled to the tandem mass spectrometry. So in this case, the timed selected reaction monitoring, which actually corresponds to the MRM or multiple reaction monitoring, and this in a dual column setup that I will explain in more details later on. So our main idea here was really to expand the lipid coverage as much as possible, even for those lipids for which we kind of lack the sensitivity, at least with the shotgun approaches, and then also to further improve actually the quantification accuracy. So finally, we've really opted for HELIC, mainly due to its advantage to separate lipids per classes, so according to their head groups. This type of separation allows for the coelution of endogenous lipids with the corresponding internal standards, and in this way, the appropriate correction of the matrix effects in the same solvent composition. And to really maximize, improve further the accuracy of the quantification, we decided to implement the Ultimate Splash 1 mixture, which has recently been released actually by the Avanti Lipids. So this mixture contains overall 75 lipid species, so with multiple representatives per each class with varied fatty acid composition. So please consider that this mixture was really designed to correct for this differential ionization and fragmentation efficiency, depending, of course, on the fatty acid chain length and the degree of unsaturation. Finally, to maximize the lipid coverage, we have implemented the profiling strategy with the initial qualitative screen. So this screen was built on the literature search yes, sorry.
Can you interrupt just for a second? Back to the last slide.
Can I go back?
Yeah, just one. Go back.
Yes.
So there's been a lot of effort to shorten this time down, in some cases using virtually no retention time whatsoever, and just pushing in everything all at once. And here you're using a relatively short retention time of roughly 3 minutes or so. Could you just brief, because I think this is an extremely important point. And why do you go towards this? What are the advantages that you see in extending it out a couple of minutes in terms of per analysis versus just doing a direct injection or virtually no separation whatsoever?
Okay, so one of the main advantages would really be the sensitivity for the detection, and then also the reliable measurement of these lipid species, which are low abundant, if you want. So this mainly refers, so if we compare directly the coverage of the direct infusion analysis with this HiLic method, you would really see that we globally cover a higher diversity of lipid species, and that especially when it comes to the different classes of phospholipids and sphingolipids.
Okay, perfect. Because I mean, personally, I don't see the 3 minutes costing you much in terms of time to get what is probably going to be much higher, much better data in terms of reliable data.
Yes, especially when it comes to the, as I said, lipids that are lower abundant. I mean, especially compared to the neutral lipids, because neutral lipids are really the most highly abundant. And yes, with DIA analysis, there is no issue to detect hundreds of these neutral lipid species. But then, yeah.
I'm sorry for interrupting. I just thought it was an important point to make with respect to, and get your thoughts on why you chose this approach.
No worries. Thanks. So yeah, here I wanted to explain why we went ahead with this initial qualitative screen, which was actually built through the literature research to include the maximal number of transitions for lipids that were actually previously reported in different types of samples. So this including the plasma, serum, but also the whole blood, different types of blood cells, and different types of tissue extracts. And so as you can see in orange on this first part of the scheme on your left, this initial screen today contains more than 2,400 different transitions actually for as many different lipid species. So these transitions, as you can see, are distributed across 4 different methods in positive and 4 different methods in negative ionization mode. And this essentially really to achieve the optimal peak definition and the sensitivity. So please consider that this initial screen is performed on a pooled sample, which is actually representative of each sample group. And then following this initial screen, the lipids that are detected across different replicates, so independently prepared or independently extracted replicates of pooled material, are actually removed, filtered according to the criteria that you can see in the middle of this slide, in order to, at the end, to be able to measure only really the robustly detected species across, in the entire sample batch. And so here you can see see how many species can be actually measured combining 2 different methods, so in positive and negative ionization mode in the NIST plasma material, and this in a total runtime of 12 minutes per sample overall. So this runtime actually was achieved with a dual column setup, so using 2 pumping systems, as you can see illustrated on this scheme on the bottom right. So we use, in our case, the Vanquish Horizon Duo UHPLC system, but this can be done, this can be set up easily on any different vendor's system. And further on, as you can see from these graphics that actually represent number of transitions across the chromatographic gradient, you will notice that in a specific parts of the gradient, the coelution is high as expected from this type of analysis that are based on the class separation. However, and despite of the fact that in a specific parts of the gradient, even up to several hundred species can indeed coelute, the good peak definition with a minimum of 8 to 10 scans was achieved even in these areas of really high coelution, which especially corresponds to this coelution of neutral lipids in the beginning of the positive ionization mode. I mean, the method used in positive ionization mode. And this was made possible thanks to this superior acquisition speed that we could achieve by using the TSQ Altis instrument from Thermo. So we could really bring the dwell time down to 0.3 or actually 0.25 milliseconds. And here on the graphics, you can see actually the peak definition and the peak shape on different representatives of different classes, which are measured in positive and then also in negative ionization mode. So overall, using this workflow, we could achieve a very broad coverage of 22 lipid classes, and we could measure around 820, exactly 820 lipid species, in this case in NIST plasma reference material. So here you can see really the coverage that was achieved in the positive ionization mode. So in positive mode, we mainly focus on the measurement of different classes of neutral lipids and also different classes of sphingolipids, and this including the ceramides, dihydroceramides, mono and dihexosyl ceramides. And recently, we have also added the acyl carnitines. Finally, in negative ionization mode, we measure essentially all different subclasses of ester and ether-linked glycerophospholipids. So as you can see, the phosphatidylcholines, ethanol amines, glycerols, serines, and inositols. And we have also recently added to this method the tissue-specific cardiolipins. Finally, as you can see, the measured concentrations, so here in the NIST plasma reference materials, span wide dynamic range of up to 6 orders of magnitude. So starting from this really low abundant hexosyl ceramides, phosphatidylglycerols, and up to this really highly abundant cholesterol esters and other classes of neutral lipids. Finally, to benchmark our method, we used the entire suite of plasma materials from NIST. So in addition to the 9050 material, we also profiled the material collected from the patients with confirmed diabetes, then also confirmed the hypertriglyceridemia, and also the pooled sample collected from the young African Americans in their 20s. And here on the right, you can see the circular plot, on which you can appreciate the differences that have been found when you compare this NIST material collected from the patients with hypertriglyceridemia to the NIST 9050 reference material. And as expected, you can see that more than 1/3 of these measured 3-acylglycerols were actually present in a significantly higher concentrations in the high TG material compared to the NIST 1950. And here I really want to make an important point that these differences in lipid composition that we can nicely see between plasma materials that are collected from different individuals can lead to significant differences in matrix effects. And you can see this nicely on the suppression of the signal of the internal standard. And so here I just presented this example that goes up to, more than actually the internal standard in the high TG material was suppressed more than 40% compared to the NIST 9050 material. And when we talk about population studies, so human studies, these differences should really be corrected with the internal standard spike if you wish to reveal the subtle but physiologically relevant changes. So this is especially relevant for the heterogeneous human populations where there is a multitude of confounding factors that actually affects or influences or contributes to the measured variability. So in human populations, you won't find these contrasting changes that we can easily and nicely reveal with untargeted methodologies in model systems. And you can actually generate a multitude of false positive and negative results if you do not correct for the matrix effects. Even more, you won't probably even be able to reveal the significant differences because they often get down below fold change 2. So they are usually comprised between this approximately 20 and up to 80% approximately. But this, of course, depends on the health or the disease state. Finally, beyond the internal standard spike, what is really important to consider is also the authenticity or the corresponding structure of the internal standard. And here, you can see the results of the measurement of 2 ceramide species with authentic internal standard, so here encircled in red, and then the analog or surrogate internal standard in blue. So these measurements were done in the context of an inter-laboratory and cross-platform ceramide trial, which was led by the Singapore Lipidomics Incubator, so Professor Marcus Wang and Federico Torta. And these ceramides were measured either using the SOP, so the standardized protocols, or any other method which was available in the lab. And as you can nicely see, the concordance of the results between labs was actually not affected by the method that was applied, because as you can nicely see, the values that are generated by the SOP group, so here in red dots, versus the values that were generated by other methodologies, so here in blue, were not significantly different. But actually the calibration with using the authentic labeled standard was the one that contributed to this, that allowed us actually to dramatically reduce the data variability. And so as you can see from these graphics, going from the 60 down to 10% only. So here you should really consider that it's only with the authentic internal standard that we can indeed correct appropriately, at least for the matrix effects. And this is very important to consider, especially when the endogenous lipids that we are measuring do not necessarily collude with their internal standards. So when we use the reverse phase methods, where we cannot possibly with one internal standard correct for the matrix effects, that will appear at different retention times in different solvent compositions, and not to even speak about the different, I mean, that this will of course cause the different insourcyanization efficiency and also the fragmentation efficiency, which will vary depending on the fatty acid chain and then also the degree of unsaturation. But for this, I mean, there is still no perfect solution, of course, simply because the standards are, I mean, all these standards are not available on the market, and it's impossible to cover the diversity of lipid species with the internal standards, I mean, corresponding internal standards. So finally, after this parenthesis on the importance of usage, I mean, the internal standards, I will now briefly share with you the preliminary data that we generated from the analysis of these first 13 batches we managed to analyze so far from the Lausanne population. So altogether, this is more than 1,000 samples belonging to 360 individuals over these 3 time points, as you can see on the scheme. So please consider that with the developed pipeline that I just presented before, we can today analyze 5 sample batches per week, so during the 5 working days, and this including the internal QCs and the NIST as the external QC. And similarly, as we do in the untargeted workflow, we have also used here the NIST, so the pooled NIST as an external quality control material to correct for the signal intensity drift over time. So mainly for this batch to batch variability. And this was done, of course, on the basis, per each, on the basis of lipid species, per each lipid species. And as you can see from the histogram on the right, after this signal intensity drift correction, which in this case was done with the simpler linear regression modeling, 790 species with the CV below 20% were actually retained for the further statistical analysis. Finally, here you can appreciate how this CVs, this analytical variability or the batch-to-batch reproducibility looks across different lipid classes. So you will notice that for all lipid classes, this average batch-to-batch or intra-batch precision is lower than 20%, with the exception of phosphatidylserines, which could barely be detected in the NIST plasma material, and this has likely then impacted, of course, the variability of their signal. Please consider here that this is why it's so important to actually, on the top of the external quality control samples, to use also the internal pooled samples if possible, not to discard some species that actually might be relevant for the population you are studying. So in this case, we kept the pieces because they can be nicely detected in the pooled sample from the Lausanne population. And finally, we went the step ahead and also beyond this analytical variability, also so far evaluated also the biological variability. So this specifically the intra-individual variability here in the light blue and the inter-individual variability in the dark blue. And globally, as you can see, the biological variability was always 2 to 3 times higher, of course, depending on the lipid class and species compared to the analytical variability. And then further on, the intra-individual variability, as you can see, was also consistently lower than the inter-individual variation. And this implies that the individual lipid profiles are indeed highly distinct and stable over time. So this made us actually explore them further using different unsupervised multivariate analysis. And so we use the TESNI, PCA, and here you can see actually the results of the hierarchical cluster analysis, which show really the distances in lipid signature, so in lipidome for plasma samples from these 20 randomly selected participants across these 3 different time points, so overall 60 samples. And so as you can see here highlighted in different colors, the lipid signatures indeed show a very high level of individuality with the samples from the same individual clustering together, and this at a time scale of 10 years. And please consider that, of course, the similar results were obtained by successive resampling of different samples from different individuals. And these results are also in accordance with the results that have been recently reported for another independent longitudinal study that has been done by the Professor Snyder group. So interestingly, they measured not only the complex lipids, but also the polar metabolites, proteins, and the transcriptome. And interestingly, compared to the transcriptome, proteome, but also the polar metabolome, the lipid signatures seem to be very highly personalized. So as a reflection really of our really chemical individuality, so that of course reflects the polymorphisms in our DNA, but also the lifestyle exposures that are unique actually for each one of us. And finally, beyond just this high individuality, we were able to nicely confirm that the plasma lipidome is also sex-specific. And as you can see from the circular bar plot, within the measured lipid signatures, more than 50% of measured lipids actually displayed very significant sex differences, so subtle but significant sex differences. So as you can see from this circular bar plot, where we plotted actually the differences of change between the lipid levels in females versus males, you will notice that the sphingomyelins and ether-linked phospholipids were found to be present in significantly higher concentrations in female plasma, whereas in contrast, the males showed higher concentrations of triglycerides and specific species of lysophospholipids. And these differences, of course, must be further investigated, but overall, we assume, I mean, we hypothesize that there must be sex hormone-driven and or the result of differential activity of specific enzymes in the metabolism of sphingolipids, etherphospholipids, and also the glycerophospholipids. So some of these, I mean, differential activities of specific enzymes involved in these different pathways have already been demonstrated. And while many of these findings are new globally on the class level, they are in accordance with previously reported sex differences in a complete health cohort, but also a very big Australian cohort, so AUSTIAB cohort, which is ongoing in Australia. And finally, just to conclude, we believe that all this, I mean, results, specifically this high individuality and then sex specificity of our lipid signatures, really call for the determination of stratified and hopefully even personalized reference intervals for all these species one day. But also they call, of course, for the further investigation and consideration of sex differences, and this not only for the treatment, so the drug dosage and clinical diagnostics, but also for the cardiometabolic risk assessment. And with this, my big thanks go really to my team in Lausanne. So a really skilled, motivated team of people. And as I will not name all of them, but you can see their names. I mean, for the sake of the time now, as you can see, we also care about our cardiorespiratory fitness and our nutrition as much as possible. Many thanks then also go to all our collaborators. So the fantastic team of Thermo Fisher, so the triple quadruple team of Thermo Fisher in San Jose, who was really supportive. The entire clinical consortium and statistical geneticists from Colaus and Complete Health and Heart cohorts. Gonzalo from Imperial College for this first statistical analysis. Martin Gira and his team at Leiden University Medical Center for the cross-validation of our method with the lipidizer. Many thanks also to Amaury from the SLING for the correction of the isotopic overlap. And finally, many thanks to all of you for your kind attention. So I will really finish now by providing you actually this glance of this beautiful view we can enjoy on the Lemon Lake here in Lausanne. And I really want to wrap up my talk with this quote from my team, which says, "Keep enjoying untargeted, but please don't forget to appreciate the power of targeted analysis." Thank you.
Okay, thank you, Juliana. So we're just going to jump to a couple of final slides, Juliana. Thank you very much. And I know it's already late, both in Asia, very late in Asia, and very late in Europe as well. So I am. Yep. Just finally say that next week we're going to go a little bit more into untargeted analyses and cover XEMS Metlin, the recently integrated XEMS Metlin, and finish up with quantitative subcellular imaging at the organelle level with Mike Kersey, who's going to be visiting from Sweden at AstraZeneca and Gothenburg. So thank you all for your attention, and sorry, it's gone a little bit over, but I thought overall it went extremely well. Thank you very much.